Thank you again, everyone, for joining us for this Legionella Rapid Response for Outbreak Prevention webinar. My name is Rony Iraq. I am the Quality Assurance Manager of Environmental Diagnostics Laboratory, and I have here with me Dr. Rajiv Sahay, who is the Laboratory Director. Today, we will be talking about Legionella, how to collect samples, and how to get quick results in order to, as the title says, get a rapid response for an outbreak prevention when it comes to Legionella. So let's go ahead and start and talk about Legionella. What is it? It is an environmental bacteria that occurs naturally in the environment, indoor environments, outdoor environments, and it was first discovered in 1976 during the American Legion Convention when many people were sick and many actually died from this bacteria. A little bit of science about it. It's a gram-negative bacteria. It's shaped as a rod. It can be anywhere from a coccobacillus, sort of like a round rod shape, to filamentous, as it says, like a filament, very long. And it's also a pathogenic and a nosocomial bacteria. Go ahead and pass it over to Dr. Sahay by asking, what makes this bacteria so important and what kind of damage does it cause?
Good afternoon to all the participants, and thank you, Ronnie. This is a very interesting topic, and I'm pretty sure several of the participants, if not all, must have heard about this bacteria and their ill effect. Well, Ronnie, you asked a very good question that why it is important, why even we are considering this as a topic of today's webinar. Well, I'm just going to give you some idea, not to scare, but to bring the factual statements about this bacteria. As you can see on this slide, it's about 8,000 to 18,000 cases per year alone is reported in the United States. I'm not including those cases which is not reported or documented anywhere, but on record, it is about that number. 90% case, as I said, is go unreported because it is very hard to pinpoint it that the symptom is from Legionella. That is one of the things. So it is not like a very pronounced. It has symptoms, but it is many times confused with some other organism too. So that is one of the reason, and obviously there are a lot of people who doesn't due to lack of awareness doesn't think about that, and it goes unreported. About 23% of all those reported cases are pretty much hospital acquired, and 77% cases are non-hospital acquired. 20% cases are travel associated in Europe. We do not have at this time data from the United States, but in the European country, about 20% of these cases are travel associated. About 10 to 20% of all the known cases basically become fatal, and people die due to this one. So this is pretty alarming situation, and we need to create more awareness so that we can respond and prevent any kind of outbreak or sort of epidemic due to this bacteria.
Okay, that sounds good, Dr. Sahay. I'm going to actually go ahead and throw up a question for everyone to answer. See that people are paying attention, trying to learn during this webinar. Legionella is a what? Bacteria, a mold, a virus, or none of the above? Go ahead and give everyone about 20 seconds to go ahead and answer that. Okay, and now I'm going to go ahead and close the poll, and we'll see how everyone did. And it looks like 100% of you got it correct. It is a bacteria. And now we will go ahead and continue on the next slide.
Well, as in continuation of that factual statements that this Legionella bacteria basically having three different kind of symptoms. The first one is called as Pontiac fever or the mild febrile illness. This is basically an acute self-limiting illness with flu-like symptoms that include fever, chill, headache. These kind of symptoms you will be experiencing during the Pontiac fever, and usually it lasts for 2 to 5 days and go away their own without treatment. Remember that I was telling you that sometimes it is go unreported. That is one of the major contributor to that phenomena. Then the other one is pneumonia-like symptom. This is more directly associated with that, and it is called as Legionnaire's disease. It shows symptom other than the mild febrile illness, like include coughs, shortness of breath, high fever, muscle agony, headaches, neurologic abnormalities, these kind of things, gastrointestinal symptoms, nausea, vomiting, watery, diarrhea, etc. These basically or usually begins after 2 to 14 days after being exposed to the Legionella bacteria. The other very common symptom is the extrapulmonary syndromes, then where you get like a pulmonary infection, and that is obviously going to have some shortness of breath and other type of infections. So these are the three major symptom you can observe once you expose to the Legionella bacteria if the strain is the pathogenic.
Okay, so now we know what it can do to individuals. Can you tell us, Dr. Sahay, what groups are at higher risk, if any?
Sure. The people having the compromised immune system or immunity is like the major high risk group in the population, and that's why it is called as immunocompromised individuals. I'm not trying to make you feel that if you get older you are subject to all kind of diseases, but it is a fact that if you are above 50 year or more, then the chances of acquiring this infection is more in comparison to the other. When you are in a community where the outbreak of Legionnaire going on, then obviously you are on the verge of getting the infection. If you are susceptible, individual with a smoking habit is more vulnerable than others, and obviously people suffering from the lung diseases such as COPD, etc., is more vulnerable in comparison to the others.
Okay. How can you tell if you might be at the onset of a Legionella infection, or what are some things to look out for?
Well, if you come to know that more than two individual or cases initially space related within a very close proximity and having similar kind of symptom without a source that they for sure say that it is coming from there, then that kind of thing is a cluster that can be a Legionella. But once you have two or more cases with the sickness or symptoms, and there also a space are within a certain a small period of time and originate from a common source of infection such as supply water, etc., then that is a form of outbreak, and then you can more often than not find out that it is an outbreak of Legionella if the symptoms begin with showing you the Legionnaire or Pontiac fever, or any symptom which you can realize in upper respiratory or things like that.
Okay. If I wanted to start an investigation, what are some of the sources that I should consider to sort of figure out if this is a cluster or an outbreak?
As you can see in this particular slide, that the very first source is the contaminated water. Some come from the soil contamination, such as potting plant and etc. If they are having infected soil, that can be spread out from there. And obviously a small percentage also come from the air contamination, especially when spraying or in very hot summer misting activity goes on and the contaminated water used in that kind of scenario, then you will be also getting Legionella spread through the air. So these are the three main sources to be considered while you're looking for investigating the Legionella.
Okay, so we're off to a good start so far. I'm going to go ahead and start a second question for everyone. How many cases of Legionnaires are reported in the USA each year? Go ahead and give everyone about 20 seconds again. Okay, I'm going to go ahead and close the question. Let's see how everyone did. And once again, you got 100% of the respondents answering correctly, Rajiv. So it looks like you're doing a good job explaining Legionella for this. So let's go ahead and continue.
So as I indicated that there is three main sources where you can get the Legionella or Legionella get spread. But the question arises that all the sources which I said you, the water is one of the main source where you can get Legionella bacteria. So the question is that where we can get these kind of water. Well, the answer is very simple. Under the natural environment, like aquatics such as pond, lake, groundwater, seawater, etc., is the source. Whereas the water can also be found in the terrestrial environment, especially like in mixture of the soil or biofilm, etc., that can also be served as a source of the Legionella bacteria. Whereas in the artificial environment, the water source mainly consists of potable water distribution system, cooling tower and evaporative condensers, swimming pool, spa, hot tub, HVAC system, healthcare facilities, hotel and ships, water park and fountains, and other indoor environment water source such as water fountain, etc., that can contain the Legionella bacteria if the contaminated water supplied to these sources.
Okay. And just to kind of clarify one more time, even though it can be found in the air and found in surfaces or soil and things like that, the most common place to test for Legionella is water sources.
Of course, yeah, because this is waterborne bacteria, in other words.
So now we have a little bit more of an understanding of Legionella. Could you break down the proactive and reactive ways of responding to Legionella in a system?
Well, as you can understand that before the problem start, if you somehow or the other have the information and you are in a position to control that, that is awesome. So this can be of two method. One is proactive method, other is the reactive method. In the proactive method, we can do very simple steps such as do a periodical test to find out whether the Legionella is present or absent in the environment which you are wondering about. And then if you have presence of Legionella, then you can proceed for a better understanding that what is the source, from where it is coming, so that you can take full control of that, so that the ill effect of that bacteria is not going to happen in future. Whereas a reactive, unfortunately, if you have a complaint and people start complaining that they are suffering from the Legionella infection and things like that, then in that case, the method or step you try to identify the bacteria, try to understand the source of bacteria. These all things come under the reactive measures, and then under these steps, like you have to again find out that whether the Legionella is present or not. You are responding the cluster or outbreak, and then the main thing under the reactive measure is going to be the investing the source where this thing is spreading out, so we can make a source cause relationship in order to manage that. And of course, once you have the situation under control, then do not forget to put a surveillance like activity in order to keep monitoring that Legionella is still a problem or it is being kept under control. So the other question arises about, like, this is the title of this webinar, the rapid response, how we can respond in a very quick fashion if we have to go under the reactive measure. So very first thing we should be start doing is the risk and hazard evaluation, that what area, how many people, how it is being spread out, if we know the source or not. If we know the source, what we can do about. We can discuss these things a little later in this webinar, but that is the first step for a rapid response. The second step is to evaluate and identify the building water system, and then that can also be supplemented by sample collection if needed, then laboratory testing to find out whether the Legionella disease is being spread by the Legionella which is in that water system or not. And then obviously if you have a positive response with these steps, then also we have to come up with a control measure so that we can manage the Legionella under control. Further elaborating the risk hazard assessment, many times people ask me question how to begin with. The very first thing, as I was explaining earlier, that we need to determine the source. So evaluating or investigating the source is going to be the first stage, because once we find out that what is the source of the Legionella, where the Legionella bacteria is being supplied, then obviously we can start exploring the causation and the other factor which is linked to the cause of the illness or sickness. Then we can also have to find out that whether it is a nosocomial or non-nosocomial. Nosocomial meaning whether it is acquired during a hospitalization, like you go for some kind of treatment and you come out from the hospital, you get your symptom better with the illness which you admitted in the hospital, but actually inherit something which is like you were not begin with. So in other words, hospital acquired infection. So you have to also find out that whether it is acquired in the hospital environment or you have acquired in your day-to-day living environment. You have to also evaluate that the illness or sickness is spreading in terms of cluster, or it is an entire epidemic, entire society or entire neighborhood is suffering from that. And then the risk evaluation is incomplete without doing the epidemiology. So find out how many people are getting sick, whether any previous episode of this kind of illness or sickness is not from that area or not. These are the other evaluation we can do or we can perform. And obviously the communication is the key or essence, so that people is not going to freak out, because if there is a problem there is a solution. So we need to communicate it properly in the society through various print media, radio, other sources of media, so that we can bring the right picture in the community or the people, so that we can manage these infection and not people get scared that what is going to happen next.
I know that was a lot of information, everybody, but I'm going to go ahead and give you a little mental break and launch a third question. Legionella is typically reported from what? Air, water, surface, or soil? Go ahead and give everyone about 20 seconds. Okay, I'm going to go ahead and close the question now, see how everybody did. And everyone is doing fantastic so far. I got 100% on the correct answer again, which was typically from water. Okay. So going Back to this. Dr. Sahay, can you elaborate on what a building water system is and how it relates to Legionella monitoring?
This is a very good picture and diagram on your slide. So the building's water system in nutshell is the source of water, from where the water is coming in the building, what are the point of use where these waters are being used in the building, and finally where these things are disposed of. So this whole complex water distribution system can also be termed as building water system. Some of the point of use you can see is given on this particular slide, such as the ponds, the cooling towers, faucets, shower head, you have a humidifier, AHU drain pan. These are the sources where the water is being utilized, and this is the point of use, or can treat it as a point of use too.
What are some of the conditions that can contribute to Legionella growth?
As you can see on this slide, that the water is a very complex, or water possesses a very complex ecological system. So it has tons of things. For Legionella, though, it is important to understand what is the pH of the water, what are the availability of the nutrition, whether there is a biofilm in this particular water or not, and the chlorine residue. I guess these 4 points is very, very important, beside the temperature, that also play a very important role to figure it out whether it is a favorable condition or not a favorable condition for Legionella growth.
Could you tell us about some of the things that should be initially determined when it comes to collecting Legionella samples?
Yeah, of course. Very first thing, you have to determine where you are going to collect the sample and try to mark those spots where you are going to take this or collect the sample, such as point of use or the water reservoir, or where you are going to select the sample. And then second thing, you have to determine how much sample you're supposed to be collecting, which I will be talking a little later in the presentation.
Okay, so now before we go on and start talking about the different methods for sample collection, I'm going to go ahead and do another question for everybody. What factors can influence Legionella growth? pH, salinity, both of the above, or none of the above? Give everyone 20 seconds again. And I'll go ahead and close the question. Let's see. All right, everyone's doing well. Got 100% correct on that one. It is the pH out of these answers that can influence the growth of Legionella in a system. So we've talked about what is needed
for testing the Legionella.
Yeah, I'm sorry.
Yeah.
We've talked about what's needed for sample collection, such as the volume, the method, etc. But can you elaborate on how to collect the actual Legionella samples?
Yes, that a very common way of collecting sample is that you collect a sample from a water source, such as potable water source. You should be collecting ideally 1 liter in a clean sample collecting bottle made out of probably propylene or the glass material. And if recently that water is treated with the chlorine, then you have to put a stabilizer that is called as sodium thiosulfate. You should be using about half a drop per liter of water to stabilize this, and then tighten the sample, and then make a chain of custody, and then send it to the laboratory for doing the testing. Now, if you are collecting the sample from the like a non-potable water source, such as cooling tower, etc., then you have to be using the same method, but you should be collecting the water either from the makeup water tank or from the slum. Try to avoid a lot of debris and things like that, but you should be again drawing the sample from the water source, as I said, and then you use the same method and you send it to the laboratory. The other one, which is like, in the natural source, such as like, the fountain, pond, etc. Again, 1 liter water is really like, good. But if you are doing from like, whirlpool or fountain, etc., a small portion of water, such as 100 to 300 ml, is also collected sometime to utilize like, for sample testing using aseptic method. Collect these sample, send it to the laboratory. The other method of sample collection is the swab, which we can use a swab to collect the sample. And what you should be doing, this kind of method is used in collecting the sample from the faucets or like, shower head, etc. The swab sample is lifted, and then the swab sample is moistened by a drop of like, water or so, like a sterile water, and then it should be placed back in the container using the aseptic method, and then it is being identified with a unique identifier, sent it to laboratory for the further processing. The other type of sample which is acceptable is the air sample, which one can pull the air sample on a specific agar if at all possible. That is called as BCYE. That is the media which is used to support the Legionella growth. One can use the Anderson sampler or any other sampler to impact the air on the culture media, and then the sample is being transported to the laboratory under aseptic condition. Typically, it is recommended to pull the air 23.1 liter per minute up to 10 minutes, because the Legionella concentration are not that great. If it is there, then if it is like, less Legionella, then you have to pull more air. But if it is a known case or known site where Legionella is known, the less volume can also be collected for testing.
So we've gone over the history of Legionella, what it is, proactive versus reactive measures. We've gone over methods of collecting. So we've got all this. Now we're sending the samples off to the laboratory. Can you go over the important methods used in Legionella testing?
Sure. The laboratory testing, there are basically 3 different kind of method is used nowadays. The first one is the non-culture method. The non-culture method uses either direct fluorescent antibody, and I will explain a little bit more on that, and the antigen identification and the rapid test method. There are a number of vendor which sells like, rapid test method, where you have like, antigen based reaction. One can just like, put the sample and let it react with the proper antigen antibody reaction happen, and it can be visualized with the color change, and that's how it is being like, studied, that whether Legionella is in that water or not. In direct fluorescent antibody mechanism, it's the microscopy is used. There's a fluorescent dye is there, which is being used to make a slide out of the collected water sample after like, going and vortexing and concentrating the sample. A portion of that is being taken on a microscopic slide and treated with the fluorescent dye with antibody, and then look under the microscope, and the fluorescing cells of Legionella can be visualized under the microscope, and that's how Legionella is identified and recognized by a trained microbiologist. So that is the first method. The other method is the second one, which is the gold standard in the industry and called as culture method. And in this method, first of all, in first stage, the recovery of bacteria Legionella is being performed from the sample which is submitted to the laboratory, either by filtration or concentration or acid treatment. These are the 3 very popular method by which the Legionella is being like, recovered from its source, and then it is being cultured on its like, media such as BCYE, buffer charcoal yeast agar media, and then later if the colony grows on those media, then it is being tested by using agglutination method, that is a confirmatory test method, to find out the species in question is Legionella or not. So agglutination test is very important to confirm the culture itself for Legionella. The only limitation of this test is that it is a little bit longer. It takes about 10 to 12 days, and there are, like, the various protocol known, and a little bit standardization is there, such as the CDC elite method is there. The ISO also recommended a method to do the culture. So there are currently two methods is being practiced. One is in short form is called as CDC method. The other one is the ISO method. Now, excuse me. The third one is the advantage of this method is that it is very rapid, and if you are in rush, then this is the method to at least get information that whether the Legionella is in that like sample or not. And it works by using the DNA amplification. What happened that the Legionella bacteria, as you can understand, it is a living thing, so it has genes. So what we do with the help of polymerase chain reaction, we multiply exponentially that gene, and with the help of molecular diagnostic using, like, various kind of dye, we monitor or we evaluate the genes present in that particular sample that can be indicative whether the organism is there or not. As I said, the greatest advantage of this method is that the time it saves tons of time, and a rapid response is really possible. At least before the culture results appear, we can, or evaluator can make a decision that how we are going to respond to a scenario, whether the outbreak or epidemic is happening. So that is called as DNA amplification method. I just wanted to give you some more information on that. As you can see that the DNA amplification method, as I said you, that, like, the gene is being multiplied, and you can see in this particular, like, slide that we put a control, and on the control, like, against the control, you see that if sample is containing the same kind of similar kind of gene, it is going to run exponentially, and it is going to give us, like, the standard curve versus their concentration. And that's how we find out that whether water is containing the DNA of the Legionella or not. Another limitation of this particular, like, technique is that, like, even if a small quantity of gene of that particular bacteria is available, it is going to show up positive. And if it shows up positive, meaning that the water somehow or the other infested with the DNA. However, sometimes those DNA are not quite enough, or not, like, quite in sumptuous quantity to form a colony on the media. So sometimes it kind of, like, end up as a false positive in a scenario where we are hunting for a viable Legionella. But nevertheless, this is a very, very important method, especially when we are running against the time, and it will give us good information to like, determine the next stage.
Yeah, and I know that here in our laboratory, Dr. Sahay, we do perform all three of these different methods that you just went over. And it's pretty impressive to see the way that the technology has sort of evolved over time, especially for this DNA amplification, using PCR, using the polymerase chain reaction. You can get these different types of results for whatever your need might be. And once you get those results, the laboratory obtained data, how can it be used in response to a Legionella outbreak?
That's a very loaded question, Ronnie. That, as I explained earlier, that there are various methods used to determine whether Legionella is there or not. Well, let's summarize this in the two major parts. If our goal is to understand presence and absence, then the result can help us to evaluate sort of Legionella present or Legionella absence. Obviously, all the three methods are going to give that information. If we are going through the DFA method, or non-culturable method, culturable method, or DNA method, we will be able to give you that result whether Legionella is there or not. So that first interpretation can be made by using any of these tests. The second one is a little bit more detailed and comprehensive test. Like once you know that you have the Legionella, then obviously question start like just questions start like going around that how much it is, uh, whether it is like a lot. And by the way, there is nothing like you can say this is less or this is more, because there are very few guideline, if any, available. But obviously if we know that how much Legionella is there, then it sort of give us pre-hand information that the quantum of Legionella or the viable cell in that particular sample is going to be harmful. As I repeat, that it doesn't matter like how small or how big the quantity is, because these are bacteria. They can multiply exponentially in a very short period of time. But it is very important to understand that if you have a lot of bacterial colony forming unit per ml, or most probable number of colony per ml, some of the method report the result in that way, that it will give you some idea that what kind of situation you are dealing with, so that you can design the further evaluation or investigation of that scenario.
Before we start our last section here of the webinar, I'm going to go ahead and launch the last question that we have. Let's see, everyone's been doing so well, so I'm sure they'll get this. What is PCR? Polycarbonate reaction, polyconal reaction, polymerase chain reaction, or none of the above? Go ahead and give everyone about 20 seconds. And I'm going to go ahead and close up this poll. And as to be expected from our fantastic audience, it was 100% correct on the polymerase chain reaction for what is PCR.
So at this point, we know that how to interpret the result, how to respond the rapid respond to a case. But many a time people ask that, okay, we understand, we find out it positive, but how we control it? That's a very good question. There are two section in which I would like to explain the control measure. First one is called as preventive action, and the second stage is going to be a corrective action. The preventive actions are those actions which is being undertaken so that this problem, first of all, is not going to start. And I know it sounds easy, but it is very difficult. Well, it is not that difficult, but you need to be very, very particular on the steps, which I'm going to just elaborate here, that there is a good maintenance on the plumbing system is recommended. If your plumbing system is not having a periodical maintenance, then there will be rusting, there will be subject of having a lot of environmental contaminant intermixing with the water, and then that is not good, because that can be leading or supporting the growth of Legionella. Routine checkup of water source is one of the other step one can take to sort of minimize or prevent the growth of Legionella in their water system. One thing I keep on telling people that avoid the stagnancy is the key. If the water is moving, then perhaps it is not going to be that dangerous for the growth of bacteria, rather in the stagnant water. So a stagnancy should be avoided in any possible manner if it is possible. Then the water temperature monitoring is going to tell you hundreds of stories on that, because it is the water temperature which is one of the supportive factor of Legionella. And about 70 degrees Celsius, remember I'm talking about in Celsius, up to 70 degrees Celsius this bacteria is going to grow. So this is another thing which we need to be monitoring very, very closely, and it will tell us a lot of a story, as I said, about the Legionella growth. So controlling the temperature is the essence of controlling this particular bacteria. Now, the corrective action, what you do once you find out or you come to know that this particular water system is contaminated with the water, then in that scenario, that very first thing, that replace and discard all the Legionella infected equipment and dump it as biohazard, so that you remove, as I said, anything in the water system such as filtering device, such as point of use, faucet, etc., and replace it as soon as possible. Treat the infected water with disinfectant. There are 2 or 3 different kind of disinfectant is known. The first one is the heat enrichment, is one of the disinfectant you can take. Then the chemical wise, you can use the halide groups such as chlorine, bromine, etc., that kind of treatment. Then there is another treatment which is like, you can use the electrical discharge method, in which you have to have certain, like, copper or certain metals, and you can have a small ionic exchange on that method in order to kill the bacteria. So the ionic exchange is also going to discourage the growth of bacteria. So these are the few methods I suggested, but remember, that is a whole another section. I'm not giving any specific dose or any specific name or anything in detail, but these are the available method which you can distinguish into part. One is the physical and the other one is a chemical. So there is a physical disinfectants and the chemical disinfectant, and obviously one should consider the environmental friendly green technology for using this disinfection.
That's the end of the webinar. We'd like to thank everyone who has joined us. If you have any questions, you feel free to send us an email, or we actually got a little bit of time here left over. If you have any questions right now, you can send us a question over the chat. Or, if you come up with a question later, you can always send us an email. We're more than happy to talk with anybody about this kind of stuff. I'm going to go ahead and put up that contact information, but first I'd like to go over that we will be attending the Florida Association of School Business Officials 52nd Annual Conference in Cape Coral, Florida, as well as the AHR Expo in Atlanta. First one is in October. The beginning, as you can see there, October 2nd to the 5th, and then the AHR Expo is from January 14th to the 16th. So you can come by, stop by, see us there, see the technologies and the equipment and methods that we use. And if you'd like to get in touch with any of our specific departments, or Dr. Rajiv himself, here's the contact information. Like I said, we're more than happy to discuss these things or help anybody out with any sort of Legionella related questions or indoor environmental questions. And once again, I'd like to thank everybody for attending this webinar. Hope you learned something. And if you have any other questions, feel free to contact us. Thank you.
Thank you very much to all the participants. And as Rony said, that we are always excited to help in any fashion we can with the Legionella and other indoor air quality related issues. Thank you, and good evening and good night.